Once we have the pyruvate,
in the first lesson,
we need to get the pyruvate back
we
over
go back
into the muscle
from pyruvate
so we can
to
keep the
glucose,
cycle
and
going.
that's just gluconeogenesis.
But I told you that
Once
pyruvate
we have
cannot
the
jump
glucose,
from liver to
the
muscle,
glucose
or from
can
muscle
jump
to liver.
from the liver to the muscle,
And then
so
once in
the
the
red
muscle,
arrows shown
we
on
use
this
slide
slide
on the left
to
in
go
the
down
liver,
from glucose to
going
pyruvate.
from pyruvate up to glucose. That is just gluconeogenesis, which you've already learned about. And then on the right in the muscle, the red arrow going down from glucose to pyruvate, that's just glycolysis. So, the glucose and the alanine that's able to move between liver and muscle to bring pyruvate across your muscle to liver. And the whole reason that we do this is because pyruvate is a carbon molecule, and we need to move those carbon molecules around to make use of certain materials in the body. So this is the reason that we turn pyruvate into alanine. It's to make use of the Cahill cycle. Now, the Cahill cycle itself refers to everything you see here with the red arrows. It's the constant conversion from pyruvate to alanine, from alanine back to pyruvate, from pyruvate to glucose, from glucose back to pyruvate. That cycle continues indefinitely while we generate glutamate and alpha ketoglutarate on the side and use them as cofactors. This is the Cahill cycle. Now, that's the reason that we would convert pyruvate to alanine.
So hello everyone, our next topic is the Krebs cycle. So the Krebs cycle is a sequence of enzyme events that are essential to cellular respiration. It is sometimes referred to as the citric acid cycle or the TCA cycle. It takes place in the mitochondria and is necessary for aerobic organisms to produce energy. Acetyl-CoA, which is produced from proteins, lipids, and carbohydrates, condenses with oxaloacetate to generate citrate at the start of the cycle. This cycle creates high energy electron carriers and ADH that contribute to the electron transport chain in addition to producing ATP. So the first step in Krebs cycle is the formation of citrate. Reaction acetyl-CoA plus oxaloacetate to form citrate. Enzyme is citrate synthase and not directly involved. Cofactor is not involved in this step, but coenzyme A is released. Explanation: in this step, the enzyme citrate synthase catalyzes the reaction between acetyl-CoA and oxaloacetate. So the reaction of acetyl-CoA and oxaloacetate to form the citrate. So the acetyl-CoA has two carbon compounds because the glycolysis to that catalyzes into two, which is the pyruvate has 3 carbon compound and the glycolysis has 6 carbon compound. So in this step, the oxaloacetate has 4 carbon compounds. So 4 carbon compounds and the acetyl-CoA has 2 carbon compounds together. So the citrate has 6 carbon compound. This stage involves a two-part process that involves an intermediary known as cis-aconitate to convert citrate to isocitrate. To create cis-aconitate, the enzyme aconitase first extracts water from the citrate. Aconitase extracts water from the citrate. And then because it gets isocitrate ready for oxidative decarboxylation in later stages of the Krebs cycle. This isomerization is crucial because isocitrate becomes more vulnerable to oxidation as a result of the rearrangement, which changes the location of the carboxyl and hydroxyl groups.
The isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and in this reaction, a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is generated. Hence, the alpha-ketoglutarate is the 5-carbon compound since one molecule of carbon is lost into carbon. In the fourth step, the alpha-ketoglutarate is converted into succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase. And in this reaction, also a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is released. Hence, the succinyl-CoA is the 4-carbon compound. In the next step, this succinyl-CoA is converted into succinate, which will be discussed in the next report.
Step 5. In step 5 of the Krebs cycle, a molecule called succinyl-CoA is converted into succinate by an enzyme called succinyl-CoA synthetase. During this process, energy is released. This energy is then used to produce ATP, which is the primary energy source for cells. ATP is important because it fuels many activities in the body, such as movement, growth, and other essential functions. Step 6. In step 6, the molecule succinate is changed into fumarate by the enzyme succinate dehydrogenase. When succinate is converted into fumarate, it releases high-energy electrons. These electrons are transferred to a molecule called FAD, turning it into FADH2. FADH2 will later help produce more ATP in another part of the energy-making process called the electron transport chain. The main goal of the Krebs cycle is to break down the food we eat and convert it into usable energy. The cycle releases energy from food and stores it in the form of ATP, which the cells uses for its activities. Steps 5 and 6 are key to this process because they create ATP and produce FADH2, which helps generate more ATP. These steps are essential for making sure that the cells in our body have the energy they help to function properly.
In step 7 of this cycle, fumarate is converted to malate through hydration reaction, where water is added across the double bond, introduce OH group. This prepares malate for the next oxidation step. In this last step, step 8 malate is converted back into oxaloacetate by the enzyme malate dehydrogenase in an oxidation reaction. Oxaloacetate, which is both the starting and the ending product of the cycle, combines with acetyl coenzyme to restart the cycle. Acetyl coenzyme fluids the cycle by pairing with the oxaloacetate, continuing the cycle process.
Hydrogen ions, in a sense, naturally move from the area of high concentration in the intermembrane space to the lower concentration in the mitochondrial matrix. However, they can only pass through ATP synthase. This facilitated diffusion drives ATP synthase to spin like a turbine. Producing ATP, it's like an incredible process, much like a microscopic hydroelectric dam, generating ATP instead of electricity as the hydrogen ions flow passively. Since numerous electron pairs are produced throughout respiration, the electron transport chain repeats the process many times, generating 30 to 34 ATP molecules for each glucose molecule that undergoes respiration.
At this point, it might help to realize that moving electrons is electricity. So, it should not be surprising that these electrons can provide energy to drive these microscopic pumps made out of protein. The electrons are then transported by the special red molecule towards their next destination. The red molecule also picks up the electrons donated by FADH2, that other electron carrier that we made during the Krebs cycle. All of these electrons are passed on to another protein complex. At this next protein complex, the electron energy is used once again to transport hydrogen ions across the membrane into the intermembrane space. Notice that we are building up a high concentration of hydrogen ions into the intermembrane space. Now, the electrons are transported to a final protein complex where their energy is used once again to pump hydrogen ions into the intermembrane space, making the concentration even higher. Now, the electrons need somewhere to go, a final destination to complete their journey.
Oxygen accepts electrons as well as hydrogen by the matrix making water. This is the reason why we breathe all day. Your cell needs oxygen to accept these electrons, where the electron transport chain will grind the halt, just like a traffic. And if the electron transport stops, you won't get enough ATP to help you stay alive and you will die. So how ATP is made? A protein called ATP synthase make a channel that allows hydrogen ions to flow through from high concentrations to low concentrations. Remember that molecules tend to move from low concentrations to high concentrations naturally in a process called diffusion.
The movement of hydrogen ions across the inner mitochondrial membrane is a crucial process in ATP production. These ions, driven by their concentration gradient, flow from the high concentration area within the intermembrane space to the lower concentration matrix. However, they can only pass through a specific channel called ATP synthase. This facilitated diffusion of hydrogen ions through ATP synthase acts like a tiny turbine, spinning as the ions flow through. This spinning motion powers the production of ATP, the energy currency of cells. It's like a microscopic hydroelectric dam generating ATP instead of electricity.
The electronic transport chain is the final stage of cellular respiration occurring into inner mitochondrial membrane. Here's the concise overview of the steps 4 and 5. So, number 4, cytochrome C oxidase. The function is the complex transfers electrons from cytochrome C to molecular oxygen, the final electron acceptor. Number 5, ATP synthase is the function of ATP synthase uses the proton gradients created by the ATP to produce ATP through the karyomosis. And then for the mechanism is the photons flow back into the matches through the ATP synthase, causing the rate and catalyze the conversion of ADP plus PI into the ATP. For the yield is produce the 28 to 32 ATP molecules per gaseous molecules depending on the efficiency. So these steps finalize the conversion of stored energy and glucose into usable ATP.